What is the earlier concept? We recall once again and we will enter into the particular topic. Meiosis. The term meiosis, it was coined by Farmer and Moore. It is one of the important bit who's coined the term meiosis. Farmer as well as Moore. Yesterday I was told usually it is happened in a diploid cells. For example, if 2n is equals to 4 a diploid sexual cell, if you take, whereas in meiosis 1 process, after karyokinesis 1, after cytokinesis 1, we will get here how many cells? 2 cells. Now, what is the major concept of the meiosis? Once the cell is subjected for meiosis, its chromosomal number reduced to half. Chromosomal number reduced to half. So, here n will become as 2. N will become as 2. So, meiosis 1, it is basically considered as the right part of the chromosomal reduced to half. Hence, this stage of the meiosis 1 is a reduction stage. After that, the cell means the 2 cells are enter into meiosis 2 process. Now we will get here how many cells from each cell means the cell is divided and gives rise to 2 cells. So like that, if you observe here, every cell is having same number of chromosomal number when compared to the meiosis 1 cell. So last finally, once the cell is subjected for meiosis, now it gives rise to 4 cells. Now the forward is 4 cells are, which state means they are haploids. So now their chromosomal number reduced to half. The statement during in meiosis process, the chromosomal number in daughter cells, it is reduced to half. The statement was given by August Weismann scientist. Noticed that chromosomal number reduced to half. At the same time, we were already discussing where the meiosis process is happened. The particular cell, it is treated as meiocyte. The cell which is subjected for meiosis process is called as meiocyte. What are the examples for the meiocytes? Meiocytes' best examples are gonads. Means, and they are usually that meiosis process, it is happened in diploid sexual cells. So in the gonads, especially it is happened in animals. When the meiosis process will be takes place during in spermatogenesis process, means here the cells are are spermatocytes. Further, which gives rise to sperms. These sperms are usually haploid. That is a concern for whose story? Male. Okay. Whereas oocytes further undergoes to meiotic division. Now they forms haploid cells, ova. It is a concern for a female. So during the formation of sperms and ovum, both are nothing but called as gonads. Here the cells are subjected for meiosis. So what are the examples for the meiosis process noticed in animals? The answer is gonad. One gonad type is spermatocytes, another one is oocyte. If we come back for planes, whereas if we take, this is the male reproductive structure, normally we will call it as stamen. In stamen, above this one, a bag-like structure is present that is called as anther. Look at here. In anther, diploid cells are present. That is two ending state. The diploid cell which is present in the anther, that is called as microspore mother cell. This is the best cell to observe that meiosis process in plants. Or otherwise, the cell is called as pollen mother cell. Pollen mother cell. Okay, we'll get more details later. For example, you are a diploid cell. Now, your name is PMC, pollen mother cell, or MSMC means microspore mothers. Once it undergoes to meiotic division, this is the symbol for meiotic division. Yesterday I already told. Now it forms how many haploid cells? Means 4 haploid cells. Now once the microspore mother cell is divided and forms 4 haploid cells, these are called as microspores. These are called microspores. These microspores, further they also called as pollen grains. These are called pollen grains. So in anther, finally which are produced? Pollen grains are produced. So the cell which is responsible for the meiosis process, that is a microspore mother cell, otherwise it is called pollen mother cell. If we take that part, fourth whorl of the flower, gynoecium. In the gynoecium, it is carpel. It is having one is stigma, another one is style. In the style, ovary. Look at here. We will get explanation later. Now, within this ovary, now the black chalk is ovule. Okay. Within this ovule, a diploid cell is present. Now, the diploid cell which is present in the ovule, that is subjected for meiosis process, that is called as megaspore mother cell. That is called as megaspore mother cell. Once it is divided, here also we will get how many haploids? 4. That 4 are called as megaspores. The 4 are called as megaspore. So identify the meiocytes in planes. One microspore mother cell or pollen mother cell which is present in anther. The another one is megaspore mother cell which is present within these ovule. Once they are divided, they forms pollen grains as well as megaspores. So what are the meiocytes? Spermatocyte, oocyte, pollen mother cell, megaspore mother cell. In all these cases, which process will be takes place? Meiosis process will be takes place. Okay. We know the familiar fact. What is this familiar fact? In the meiosis process, when the diploid cell subjected for reduction division, here half of the chromosomes is concerned for father, half of the chromosomes is concerned for mother. Now they are nothing but called as homologous paternal chromosomes, homologous maternal chromosomes. We will discuss. We know the process. What is the process? Cell cycle. In cell cycle, the first phase is which phase? Interphase. Okay. In interphase, what happened, ma? One is gap 1 phase, another one S phase, another one gap 2 phase. After that interphase only, then the cell is enters either mitosis or otherwise meiosis process. For example, this is the completion of a interphase cell. Very clearly nucleus, nuclear membrane, nucleolus, chromatin as well as all cell organelles is formed. For example, if you are having here 2n is equals to 4 chromosomes. If this cell, it is concerned for growth of the organism, now the cell is subjected for which process? Mitosis process. In mitosis process, we discussed one is prophase, metaphase, anaphase, and telophase. Of course, that cell is not involved in the growth. It is responsible to maintain the chromosomal number constant. Now, it is enters into which process? Meiosis process. In meiosis process, one process, it is meiosis 1 process. The another process is meiosis 2 process. In both these stages, nucleus division will be takes place, cytoplasm division is also takes place. So, further these meiosis 1, it is classified into prophase 1, metaphase 1, anaphase 1, next telophase 1, next the division of cytoplasm, cytokinesis 1. Okay. The first meiosis process will be takes place. Now we will get how many cells here? 2 cells. Their number is reduced to half. 2n equals to 4. Here n is equals to 2 and next meiosis 2 will be takes place. In the meiosis 2, the phases are prophase 2, metaphase 2, anaphase 2, as well as telophase 2 and cytokinesis 2. So at last, finally, we will get here how many haploid cells? 4 haploid cells. This is the concept. So when a cell it is subjected for meiosis process, before in the interphase stage, what happened? Look at here. Now I would like to take first, this is a diploid cell. It is 2n in state. Now in gap 1 phase, okay, in gap 1 phase, what happened? Some amount of chromosome, some amount of DNA is present. Now it is noticed as 2C. When S synthesis will be takes place, what is the characteristic of S phase? In the characteristic of that S phase, we can notice a process. What is the process? DNA replication process. In the DNA replication process, now the amount of DNA is the same when compared to with the G1 phase or doubled. It will become doubled. For your understanding, here I would like to take 2n is equals to 4 chromosomes is there. Okay. Out of 4, yesterday I was explaining, in every cell 46 chromosomes is there. Out of the 2n 46, 1n is equals to 23. That is a concern for father's sperm. The another n equals to 23. That is a concern for mother ovum, means ovum as well as sperm. If you take 2n equals to 4. Now 1n is equals to 2 chromosomes. They are concerned for male. They are called as homologous paternal chromosomes. This word you have to be remember continuously. It will come homologous paternal chromosome. Another n is there. That is also how many chromosomes? 2. Now the remaining 2 chromosomes, half of that one, it was a concern for mother, means female. They are called as homologous maternal chromosomes. Homologous maternal chromosomes. Term is there, homologous. Homologous chromosomes means the chromosomes which are similar in their size, similar in their length, almost all similar in their shape, similar in their size, similar in their shape, similar in their position of the centromere. Look at here. We know this is the chromosomal structure. If you observe, a round knobbed structure is present. It is called as centromere. On both sides of this centromere, which are present, chromosomal arms are present. These are called chromosomal arms. Now, this is another chromosome. So, these are homologous in nature. Means these chromosomal number means here their position of the centromere is the same, their length is the same, their shape is the same. Now, hence it is called homologous paternal chromosome if it is concerned for father. Next, another one is homologous maternal chromosomes. So, which are similar in their size, which are similar in their length, which are having the position of the centromere is same. almost all they are having the same type of genes. Look at here. Now I would like to take here an example. Here a gene is there. Here gene. Here gene. Here gene. Here their genes is also same. But in what way they are different? Their alleles are different. A-L-L-E-L-A-S. Alleles. Allele means the alternate expression of a gene. Alternate expression of a gene. For your understanding, I would like to take one characteristic. What is that characteristic? Color. Color is the characteristic. Okay. This color is under control of a pair of genes. Now, for example, we would like to say one color is black. Of course, if it is not black, now we used to say here which one? White. What is the color? Black. What is the color? White. Alternate expression of a characteristic. For example, here this is the paternal chromosomes. Now you think so. This is paternal means male. This is the maternal female. Their size is similar, length is similar, centromere position is similar. All they are having which one? Same kind of genes, but their alleles are different. For example, now I would like to take here a gene is there. Now that is capital T. Here also which is there? Capital T. Here the alternate expression small t. The alternate expression here small. What is that T here? Means, for example, here we used to discuss about height of the plane. What is the height of the plane? The plane is too much of height, 6 feet height. Now we used to say that characteristic is height expressed as tallness by alleles. Okay, what are the alleles here? One is capital T, another one is small t. This is present in, for example, homologous paternal chromosome. These are present. If you take that homologous maternal chromosome, height is very short. That is described as dwarf. For the dwarf condition is also decided by two alleles. They are small t and small t. Genes are the same. Their expression is different. So, homologous chromosomes means the chromosomes which are similar in their size, shape, position of the centromere, having the same genes. Okay. For example, you and your brother exactly same in color, same or having same kind of attitude, and you and your sister having same kind of hairstyle, both are having same kind of skin tone. Your face is never matches with any other person throughout that country. So why these variations are takes place means there is exchange of chromosomal material in between these non-sistered chromatids that we will discuss. Are you clear? The homologous paternal and homologous maternal. For example, here I am showing. Okay, total 2n is equals to how many chromosomes means 4 chromosomes. One chromosome, another chromosome, these two. These two, the chromosomes are similar in their size, similar in their shape, and now the round mark is which one? Centromere. These two are derived from father. Now they are called as homologous paternal chromosomes. These are homologous maternal chromosomes. This thing it have to be remember. Homologous. As the time passes on, what happened? At the time is passes on, slowly in S phase, the number of chromatids is increased. It means, look at here. This is the G1 phase, centromere. This is the chromosomal arm. Look at all of you, observe the concept. Okay. Now, within this one, who are present, one DNA molecule is present. As the time is passes on, observe here, the chromosomal arms are longitudinally splits. When the chromosomal arm is longitudinally split, now they forms chromosomal parts. These chromosomal parts, whenever it is divided, now these are nothing but called as chromatids. Chromatids. Now, after completion of the G1 stage, next which stage will be takes place? S phase. What happened in S phase? Come and tell me. Once that DNA molecule is there, once you are divided, we will get here how many DNA molecules? 2 DNA molecules. Now you are one homologous paternal chromosome. Now you are having which one? 2 DNA. For example, this is for paternal chromosome. Next, another chromosome we have to take. This is the maternal chromosome. Here also how many DNA molecules? 2 DNA molecules. Once they are divided, now they forms here which one? 2 chromatids. In the each chromatid, how many DNA molecules are present? In the earlier case, now the number of the DNA molecules is 2C. Now in the S phase, now the number of the DNA molecules is double, 4C. This you have to be removed. Listen carefully. I would like to take an example of now, this is the homologous chromosome, paternal one, father one. It is chromosomal one. Now, the center round mark is centromere. The center round mark is centromere. Okay. Now, during this process, how they will be formed means, if you take, this is the chromosomal arm. Okay. Now, within this one, this is the centromere region. Now, this is the home DNA molecule. Okay. After that, what happened? In S stage, the chromosomal arms are longitudinally split. Now they forms a chromatid. Observe the board. Sir, this and this is a chromatid means that is wrong. This is one chromatid. This is another chromatid. Now here who are present? One DNA molecule is present. This is which one? Another DNA molecule. Are you got the difference? Okay, so this is earlier stage. This is the chromosome homologous one. Whenever the synthesis will be takes place, now the amount of the DNA will become double. Same. This is for homologous paternal chromosomes. Another one for maternal chromosomes. So after that, what happened? We will start the exact process of meiosis. So the journey of the meiosis will start now. All of you listen carefully. So after interphase means gap 1, S, gap 2. Now I would like to write here 2C amount of the chromosomal number, amount of the DNA is double, further it will be carried. After that, which process will be takes place? Meiosis 1 will be happened. Here I would like to take an example of a diploid cell 2n equals to 4. Okay. This meiosis 1 process, further it is classified into 4 stages. One is prophase 1, another one is metaphase 1, another one anaphase 1, the last one is telophase 1. During the meiosis 1 process, first the nucleus is divided. Division of that nucleus is called as karyokinesis. Karyokinesis. First nucleus division will be happen here. Hence we will write karyokinesis 1. After that, division of the cytoplasm, that will be treated as cytokinesis 1. That is meiosis 1 story. If we take that first stage of that meiosis 1, now you think so. This is our friend interphase diagram. This is the interphase diploid cell. Okay, nuclear membrane is present, nucleolus is present. Here, who are present? Chromatin is present. Which is present? Chromatin is present. That is the thing. When prophase 1 is start, look at here. Next, the prophase 1. Prophase 1, it is a very lengthy process. That is a very complex process. Hence, the prophase 1, it is classified into 5 substages. One is called as leptotin. Another one is called as zygotin. And the third one is called as pachytin. And the fourth one is called diplotin. And the fifth one is called as diakinesis. When all these 5 subphases are completed, now who is completed? Prophase 1 is completed. Now we will start our journey. Okay, now the first stage of the prophase 1 is called as leptotin. Okay, this is interphase cell. When leptotin is start, what happened? Characteristics we will write here, side. Okay. The first foremost thing, when prophase leptotin is start, what happened? The nucleus size is enlarged. Nucleus size is enlarged. This is the nucleus. Now, their nucleus size is enlarged. Gradually, nucleolus size is also enlarged. That is the second point. Nucleus size is enlarged. Second point. Whereas in interphase, are you observing any clear-cut chromosome here? No. It is available as a color thread-like structure. So T means thread-like. So what happened? These chromatin will become as short and condensed, short and condensed structures. Now, the process where the chromatin, which is short and condensed formation, that is called as spiralization. Now, which process will be takes place? Spiralization. So here some amount of chromatin is there. I used to say, for example, 2n is equals to here how many chromosomes means 4 chromosomes. Now some amount of that chromatin, look at here. This is the chromatin. It is converted according to that given picture. It is converted into a pair of homologous. I would like to write short for. Now the black chalk is homologous. For example, whose you are paternal chromosomes. You are paternal chromosomes. Some amount of that chromatin, it is converted into which one means homologous maternal chromosomes. Homologous maternal chromosomes. Come on, tell me. I would like to say here 2n is equals to 4. 1n is equals to 2 consent for father. 1n equals to 2 consent for mother. So, what point you have to be remember? In leptotin stage, nucleus size is enlarged. Chromatin is condensed and formed as chromosomes. Now the chromosomes are homologous paternal chromosomes and maternal chromosomes. For example, if you take, this is the maternal chromosome, okay, or otherwise it is a paternal chromosome, whatever it may be, look at here, whatever it may be. On these chromosomes, a small beaded structures are present. Small bead-like structures are present on these chromosomes. These are called as chromomeres. Chromomeres. The last line. The last line in the prophase I recall. They are, for example, a function is going on. Boys is there, girls is there. Okay. Now the principal is came. What happened? All the boys has to come right-handed side. All the girls has to be come to left-handed side. What is meiosis in one line means? Exchange of looks, marriage, children, apply for divorce. That is the meiosis process. We will discuss. But divorce is not easily sanctioned because of children is there. Who will be shared? One has to be shared, both has to be shared. The process is there. Like that, the meiosis process is also same. At the end of these leptotin stage, look at here. Listen carefully. This is the homologous paternal chromosomes. These are homologous maternal chromosomes. They are randomly present. At last, finally, at a particular point region, all the homologous paternal chromosomes are one side. All the homologous maternal chromosomes are one side. Here also, how many chromosomes? 2n is equals to 4. What happened here? Chromatin is converted into chromosomes. Now the chromosomes are here two types. Means they are homologous paternal and maternal. Now the homologous paternal and maternal, all they are joined at a particular place. Now it seems to be the arrangement of the chromosomes, that flowers arrangement in bouquet. We can observe that bouquet. What is the style of that bouquet? It is almost all like this fashion. Now these are the flowers. Okay, all that pedicels are present at one fashion. Hence, the leptotin stage, it is otherwise called as Bouquet stage. Bouquet stage. So, this is about leptotin. It is just a film introduction. Okay, now hero is coming in one side, the heroine is coming another side. Same thing. So all the paternal chromosomes are one side, all the maternal chromosomes are other side. Both are joined at a particular point. Hence, it is called as Bouquet stage. So first in meiosis 1, in prophase 1, leptotin process is completed. Now the nucleus size is enlarged, chromatin short and condensed, it will become spiralization. Now the amount of the chromatin is equally distributed for homologous paternal and maternal. All the homologous paternal and maternal, they are distributed somewhere within the nucleus. But at last, finally, all they will become convergent. Convergent means come close together and arise from a particular point. That is called as convergent. Look at here. This is the convergent. This will become which one? Divergent. So all they will come. All the boys are one side, girls are one side. Paternal chromosomes are one side. Maternal chromosomes are one side. So till to that, I will post in the group the meiosis introductory part, homologous chromosomes definition, along with the part 18 stage. And tomorrow we will discuss leptotin completed, zygote and part 18. We recall that concept tomorrow once again. We will start with the zygote.